Welcome, everybody. Thank you very much for joining us tonight. My name is April White, and I am a Great Lakes Program Officer with Environment and Climate Change Canada. This evening, the Detroit River Canadian Cleanup Committee is hosting a virtual open house to share study findings related to the populations of phytoplankton and zooplankton in the waters of the Detroit River. To get an idea of who is in the audience tonight, I'm going to open up a poll to get a sense of who we're presenting to this evening. Oh, that's great. Great, it looks like we have quite a diverse group. Thank you for participating in that poll. You may have noticed that your videos and your microphones are muted. We're doing this throughout the webinar this evening, but we do welcome questions that you may have for our presenters. These questions can go in the Q&A box, and we'll go through them at the end of the webinar. Any questions relating to the committee's recommendation to designate this beneficial use impairment as not impaired can go into the chat box. This will just help us manage questions and comments regarding the recommendation. For those that are not familiar with the Detroit River Canadian Cleanup, it is a community-based partnership between the federal, provincial, and municipal governments, local industries, researchers, environmental organizations, and citizens working together to protect, restore, and enhance the Detroit River, which was designated as an area of concern in 1987 due to severe degradation of the aquatic environment. This was caused largely due to industrial, urban, and urbanization. The degraded water and aquatic ecosystem led to the impairment of beneficial uses by people, fish, and aquatic wildlife. The Detroit River was one of 43 areas of concern identified around the Great Lakes. To address the impaired beneficial uses of the Detroit River, a remedial action plan or cleanup plan was created. The plan details actions needed to restore the impairments and ultimately restore the river's aquatic environment. The remedial action plan, or RAP for short, is coordinated and implemented by the Detroit River Canadian Cleanup Committee, your hosts for this evening. As I mentioned earlier, we're here tonight to share with you the findings of research that has been undertaken to assess the status of phyto and zooplankton populations in the Detroit River. The degradation of phytoplankton and zooplankton populations is one of 14 beneficial use impairments an AOC can have. In fact, on the list of the 14, it's number 13. Lucky 13. Now I'd like to welcome Jackie Saran, the local remedial action plan coordinator for the Detroit River, to provide some more background information on the remedial action plan for the Detroit River and to highlight some previous plankton research that has been undertaken to determine the status of the populations for the Detroit River.
Awesome. Thank you so much, April. And once again, thank you so much for joining us. So I'm just gonna, as April said, provide a brief background and overview of the remedial action plan, and then specifically go into the history of BUI number 13, which is the phyto and zooplankton beneficial use impairment, or BUI as we call them. So let's get started. So I wanted to start this presentation by showing you some historic photos of the Detroit River. So through the construction of shipping channels, industry emitting pollutants, and the loss of habitat, the Detroit River became notoriously polluted. So you can see here on the left, there's a bunch of oil that has spilled into the river, and you can also see a few other photos that indicate this. And then in the bottom middle is what the Windsor shoreline used to look like downtown. So you can see that we've made, for those of you who are familiar with it, a lot of progress towards greening up that space on the shoreline of the river downtown. And so in the 60s and 70s, the people in this area and the media that were reporting that Lake Erie was dying. So there were huge mats of thick green algae that you can see in the left there, and fish were washing up dead on the shore in masses. And so Lake Erie wasn't in fact dying, it was just very sick. And so this public outcry over the algal bloom problem in Lake Erie caused politicians to take notice, and they decided to make some changes. So several important policies came into play soon thereafter. So the US Clean Water Act was amended to prevent water pollution, and the Canada-US Great Lakes Water Quality Agreement was signed by Richard Nixon and Pierre Trudeau, mostly in response to phosphorus enrichment of waters and burning rivers. So it was signed first in 1972, amended in 1978 and 1987 to include AOCs. And so the goal of the Great Lakes Water Quality Agreement is to restore and maintain the chemical, physical, and biological integrity of the water of the Great Lakes Basin ecosystem. And so Detroit River in 1987 was identified as one of those original areas of concern. And it's unique in that it is a binational area of concern as well. So it's an area of concern both on the Canadian and the American side. And each area of concern establishes a remedial action plan or cleanup plan to clean up each area of concern. And so the DRCC, the Detroit River Canadian Cleanup, with its partners, help implement this cleanup plan. And so, as April mentioned, the Detroit River was considered an area of concern because one or more of the above beneficial uses was considered impaired. So you can see under the Great Lakes Water Quality Agreement, there were 14 beneficial uses identified. And so in order to be an AOC, you have to have one or more of these to be considered impaired. And so the Detroit River offers several ecological, recreational, and economic beneficial uses. And when something interferes with the enjoyment of a water use, it is called a beneficial use impairment. And so there's several stages to a remedial action plan. And so just to give a brief history of the Detroit River Remedial Action Plan, so there's stage 1, which determines the severity and underlying causes of environmental degradation. So that was completed in 1991, and you'll note that that was actually a binational stage 1. So originally Canada and the US were going to develop a cleanup plan together, but eventually they separated. And then stage 2 was created in 2010. And so stage 2 has goals and recommended actions that will lead to the restoration and protection of ecosystem health. And those are identified as well as actions to improve those beneficial uses. And then we also put out a pathway to the listing document every year. And this outlines our actions for each year and what we plan to do. And it's basically our work plan. And so in 2010, they used scientific studies and data to assess the status of each beneficial use. And you can see here the red ones were considered impaired, yellow required further assessment. So when a BUI required further assessment, there was insufficient data to make a status determination. So that's why they were classified as required further assessment. And then you could also be green, which means it's not impaired. And so today we're going to focus on the degradation of phytoplankton and zooplankton populations, which is the 13th of the 14th beneficial use impairment, and it was classified as required further assessment in the 2010 stage 2 report. And so I just wanted to provide a brief overview. Mark will get more into this in depth, but just what are plankton? So plankton are small organisms that live in the water, and they are grouped into 3 general categories. So there's phytoplankton, zooplankton, and microbes. And plankton are an important food source for fish in the Detroit River. So phytoplankton consists of microscopic plants that come in many forms. They make energy from the sun, similar to land-based plants. And an example of a well-known type of phytoplankton are diatoms. And then we have zooplankton, which are a bit larger, tend to be a bit larger than the phytoplankton. And they range in size from microscopic organisms such as water fleas down to smaller organisms like mussel larvae called veligers, which you'll hear Mark reference in his presentation. And zooplankton feed on phytoplankton and smaller zooplankton. And then we have microbes. So these are the smallest single-celled organisms, including bacteria. So certain bacteria can be a problem in aquatic systems. I'm sure we've all heard of the detrimental effects of E. coli, which results in beach closures, but most of them are a natural part of the ecosystem. And so I wanted to provide a history of the BUI number 13 phytoplankton and zooplankton for the Detroit River. So in the stage 1, report, it was actually considered to be not impaired. As well as in the 1996 Remedial Action Plan Update report, it was also considered not impaired. But then in 2006, Lenny and Hafner in their report proposed that the status be changed to required further assessment due to limited information about the composition of Detroit River phytoplankton and zooplankton, which is why they determined that it should be classified as required further assessment instead of not impaired. So then in 2010, they took the recommendations from 2006 and they ended up classifying this BUI as require further assessment. So that means we had to do some more studies to determine what the status of phytoplankton and zooplankton was in the Detroit River. And so each impaired or requires further assessment BUI was given delisting criteria. And so this criteria is what needs to be met in order for this BUI to be considered not impaired. So number 13, the degradation of phytoplankton and zooplankton populations will be considered not impaired when the composition and relative abundance of phytoplankton and zooplankton of the Detroit River reflect that of Lake Huron and therefore represent primarily oligotrophic/mesotrophic conditions. So just to give you an idea, oligotrophic and mesotrophic conditions are have medium to low levels of nutrients, tend to have clear water and some aquatic vegetation, but not too much algae or too many nutrients. And so I wanted to speak to some of the past studies that we've done on the river looking at phytoplankton and zooplankton. So there was a study in 2007 by the Great Lakes Institute for Environmental Research at the University of Windsor. And the objective was to determine if the composition and relative abundance of species of phytoplankton and zooplankton in the waters of the Detroit River changed from upstream to downstream. And so as a result of their studies, they found that phytoplankton abundance in the Detroit River was low, and they were shown to be indicative of oligotrophic and mesotrophic conditions that were representative of the upstream waters of Lake Huron. So that there was no impairment based on this study. And so then in 2014, the Canadian Centre for Inland Waters looked at 65 archived plankton samples. They were collected during different surveys and were analyzed. And so the samples showed no evidence of any degradation of phytoplankton and zooplankton communities. And then Glear in 2015 went out again on the river. They sampled in Lake St. Clair, which you'll see at the top right of the map, near Pesh Island, and then again near Amherstburg at the outflow of the river. And so the objective was to examine spatial and seasonal changes in the zooplankton community structure of the Detroit River area of concern and in Lake St. Clair. And so it was concluded that the overall quality of zooplankton communities in the Canadian waters of the Detroit River AOC did not differ from its nearest upstream water body, which is Lake St. Clair. And so as such, it was recommended that the zooplankton, specifically a community component of BUI 13, be considered not impaired for the Canadian waters of the AOC. And so we have these three studies that were done previously, but while these studies indicated no impairment in either of the plankton communities, a comprehensive study to evaluate both plankton communities and microbial communities collectively was recommended by agency experts. So that brings us to the study done by the Department of Fisheries and Oceans Canada in 2019. So from July to November 2019, scientists from the Department of Fisheries and Oceans Canada sampled 8 sites in the Detroit River to assess phytoplankton and zooplankton community composition in the water column for BUI 13, and you can see the photos here. So now I would like to introduce Mark Fitzpatrick, one of the lead scientists from the Department of Fisheries and Oceans Canada, to describe what they examined and how the results are being used to support the redesignation recommendation by the Detroit River Canadian Cleanup.
Okay, well, thank you very much, Jackie. I'll just get the screen loaded here. Okay, how does that look?
Looks great.
Okay, well, thank you so much. You know, it's it was quite a, I was really actually kind of happy to get to work on this project. My wife and I for about 3 years lived in Windsor near the foot of Crawford Avenue, and we spent a good deal of our time walking along and cycling along the river. So this was really personal connection for me to be able to come back and do this work. So I was quite excited about it. So for today's talk, I'm going to go over a few things. Fortunately, Jackie and April have gone through a lot of these, so hopefully I can get through them a little bit more quickly. But I did want to just talk a bit about planktonic food webs, and Jackie gave a great introduction to BUI 13 and some of the objectives of the study. I'm going to provide a few comparisons of the Detroit River with some other areas of concern in the Great Lakes and some open waters of Lake Ontario, which obviously aren't part of the AOCs. We'll come back to the key findings from the 2019 study that we did, and then I'll wrap this up. So, so what is a food web? I mean, I'm sure you're all familiar with kind of the classic food web pyramid. You have phytoplankton at the bottom. They photosynthesize, they generate the food that zooplankton then eat, and smaller fish eat zooplankton and bigger fish eat smaller fish. And that's kind of your classic food web. But as Jackie indicated, we also have other microbes in there that kind of affect these things. I did want to include some more pictures of some of the things we're looking at. The phytoplankton, the term itself means wandering plants. They are plants and they photosynthesize and they do provide food. Some of the microbial organisms include more tiny phytoplankton, which we're calling picoplankton, bacteria, heterotrophic nanoflagellates, which are those, they kind of look like planets there in blue with the tail on them right around here, and you can see they have a bit of a tail. They tend to eat bacteria and picoplankton. We have ciliates, small single-celled organisms, and then we get into the larger zooplankton. That term means wandering animals, and they feed on phytoplankton, on bacteria, and sometimes they compete with H&F for food. But they also provide an important food resource for the fisheries, and really that's what this BUI is getting at and why we really need to care about these things. So there's a lot of ways to look at a food web, and we've come up, and there are all kinds of different schematic diagrams and everything, but I want to share with you one that was drawn, that my summer student came up with a couple of years ago, who I think really nailed it. I just think this is such a great picture showing how a food web works. That was Jessica DeForge was her name, and this was her impression of working with us over the summer. But she even, not just the big fish and small fish, but she got the picoplankton and the bacteria and the flagellates and the zooplankton, and she got everything there. I just think it's such a wonderful representation of the food web. Again, Jackie talked about this, where we're at in terms of requires further assessment. Some of the issues we identify that have come up with the current delisting criteria really, as they were proposed, was that there's been a lot of confusion between this BUI and BUI 8, which is eutrophication or undesirable algae. In terms of what's happening, what happens to the planktonic communities, there's really no reason you would expect them to look like Lake Huron when you're about 100 kilometers downstream. It doesn't really account for things that'll happen to them along the way, or what other tributaries might do, or just a lot of things happen in that, can happen in that, over that distance. And so we want to make sure that we encompass all these things in moving forward in delisting, in possibly delisting the BUI. So our objectives, of course, were to provide a baseline assessment of the phytoplankton and zooplankton communities. So that, of course, as you well know now, includes the microbial food web, the bacteria, the picoplankton, the H&F and ciliates. We did specific measurements of primary productivity, which is algal photosynthesis using radioisotope tracers, and also bacterial productivity, and then a variety of physical and chemical measurements, nutrients, and things like temperature and flow rates and everything else. But the focus really, the directive here and the focus really had to be on the Canadian AOC. What's happening there, and is there a potential impairment of the food web that is actually occurring within the AOC? And in order to do this, we looked at a number of parameters, but we were looking for nearshore and offshore differences and upstream and downstream gradients. And I'll get into why in a few minutes. But and again, to emphasize that there's more to this than just looking for possibly algal blooms or eutrophication. It's not about that, it's about food web function. But before I get into that, I thought I'd show first some comparisons with other areas of concern in the Great Lakes, specifically Toronto Harbor, Bay of Quinte, and Hamilton Harbor, which all have this impairment under BUI 13. I also included the Niagara River, another riverine environment, which was recently found. They recently decided not to change the status to not impaired, and we've got a site in Lake Ontario as well, just to give you, just to show you what things look like across the Great Lakes and across a spectrum of environments. Again, April and Jackie introduced the AOC program and the RAPs. So, you know, we'll be looking at sites, we'll be comparing the Detroit River with some other sites around Lake Ontario, from Hamilton Harbor in the west and Toronto Harbor to the Bay of Quinte in the east. So, you know, as you well know, there's been a phosphorus abatement program in place since the 1970s, since the Great Lakes Water Quality Agreement, and we've been trying to regulate phosphorus levels in these environments for a long time, and they really are, they really do have an enormous effect on algae or phytoplankton growth rates. And one of the basic ways to characterize ecosystems is just to look at some basic nutrients, in this case, phosphorus and nitrogen in the form of bioavailable in the form of nitrate. Chlorophyll, of course, is a pigment found in all plants, and it's especially, and it's used commonly as an indicator of the amount of phytoplankton in the system. And basically what I want to show here is like the Detroit River in terms of total phosphorus. The average from May to October was about 15 micrograms per liter. So that's very similar to the offshore waters around Lake Ontario. In the Bay of Quinte and Hamilton Harbor, it's a lot higher. You're like 35 to 40 on average. Those are eutrophic environments. Those are environments subject to algal blooms. And nitrogen, the bioavailable nitrate, again, is similar to the open waters in the Great Lakes in Lake Ontario. It's not extreme at either as it is in potentially eutrophic environments like Hamilton Harbor, where it's nitrogen enriched, or Bay of Quinte, where there's nitrogen depletion because there's just too much photosynthesis going on and using it up. And of course, the chlorophyll, again, a measure of the amount of phytoplankton. You can see it's very low. So, you know, it's even actually lower than what we saw in Lake Ontario at about, you know, we're averaging like 1.5 micrograms per liter. So it's very low and sort of consistent, unlike the other more extreme environments. So we'll sort of look at the main components of the food web: phytoplankton, zooplankton, and microbial loop. And this slide's ridiculously busy. But I think what I want really to take note of in all of these cases is in the Detroit River is just how low the biomass is compared to some of the other environments along the lake. That's true for phytoplankton, that's true for zooplankton, it's true for the different components, the bacteria, APP and HNF, and the microbial loop. It's just so much lower. So, you know, it's not a question of whether this system is eutrophic or oligotrophic. It's more a question of what, you know, why is it so much lower? And, you know, and is, you know, is it so low that it's maybe there's not enough energy to support the food web? To try and answer that question, we also did measures of primary productivity or algal photosynthesis. That's this top graph here. In general, as you might expect, in the Detroit River, of course, is nothing like what we see in productive algal bloom, in the productive environments where they have algal blooms, like the Bay of Quinte or Hamilton Harbor. It's much more in line with offshore Lake Ontario. In fact, it's actually a little bit higher, and actually a bit higher than what you might expect given the phytoplankton biomass. There's a couple of reasons for that. Of course, one is we did them in the main, one of the reasons, of course, is we did them in the lab the next day. So in the river itself, you know, the river is moving, things are flowing, things are getting pushed around. It's a pretty inhospitable environment for phytoplankton, so they're not necessarily doing a lot there. It's a very tough environment for them. But when you take them out of those conditions and put them in the lab and give them optimal conditions, they're not being pushed around by the current. Temperature and light are just perfect. They do really well. So what these really show is that you do have a viable phytoplankton community there. They're working as they should. And at the same time, the productivity is not excessive. We also did bacterial growth experiments as well. You can see the main thing to look at is Toronto Harbor. That's, in terms of bacterial growth rates, that's pretty much the highest I've ever seen anywhere in the Great Lakes. So good thing is we're nowhere near that. You know, they're a bit higher, but again, the actual amount of bacteria was low, but they did respond in the lab under ideal conditions. So I like to look at food webs in terms of organic carbon. I call this the fish food slide. You can think of the carbon as kind of like the food in your refrigerator. It's what's there at the time, but it's subject to being eaten and also subject to, you know, going out and buying groceries and being replenished. Here I'm comparing the Detroit River specifically with the impaired AOCs, Bay of Quinte, Hamilton Harbor, and Toronto Harbor, which have that impairment of BUI 13. And they look very different. You can see in the Detroit River that about 80 to 85% of the organic carbon is coming from the phytoplankton. It's very much driven by the autotrophs. Whereas in the other AOCs, the microbial signal, especially the bacteria and H&F, is much stronger in the carbon budget. So I think that's a very important difference right there. And, generally a very sort of good sign about the state of the health of the Detroit River. So let's come back to the 2019 study and the work we did and the comparisons looking at nearshore and offshore studies. So the question really is what's happening to the planktonic communities within the AOC. And we did look at these things through nearshore and offshore gradients, as well as upstream and downstream gradients, and used a variety of statistical approaches to consider significant differences. And with that, the question is, are the observed changes consistent with expectations for a riverine environment? And if the answer to that question is yes, then we really don't have an impairment. So some examples of that might be that we have expectations that zooplankton biomass will be reduced from upstream to downstream. It'll be a lot lower downstream than it is upstream. That's very typical in river environments all over the world. It's and another one we have, another example is that we might expect to see higher phytoplankton biomass in the nearshore versus the offshore because you have reduced currents. They're able to photosynthesize a little bit better. They build up some biomass. But the point underlying this is that you have to very carefully consider the function of the ecosystem when making these sorts of assessments and making these sorts of judgments. Alternatively, the question then might be, are the observed changes of a nature that can only be explained by anthropogenic impairment? Ken Droulard at the University of Windsor really pressed me to come up with some examples of what an impaired system might look like. And I want to stress again that these are just hypothetical examples. This is, these are entirely hypothetical, but some examples of what an impairment might look like would be seeing bacteria-laced urban runoff contaminating nearshore waters of the AOC, which would result in more elevated microbial activity, such as bacterial productivity. You'd see it in H&F biomass and ciliate biomass and less zooplankton. It could be something like sewage effluent leaking, resulting in an uptick of phytoplankton and bacterial productivity, which would be coupled with phytoplankton and microbial biomass increasing downstream. In fact, we observed this sort of situation last fall in Hamilton Harbor. So it's not overly far-fetched. And then another example might be agricultural runoff, say from the River Canard, resulting in increased microbial activity downstream, where there is in general less zooplankton biomass. So those are just examples of what we might find if things are bad. So Jackie mentioned a bit about the study at the beginning. So we looked at 8 sites from Lake St. Clair down to Amherstburg. And we did this survey during May, July, September, and November. So we really captured the spring, summer, fall seasonal gradient. And we had nearshore, offshore pairings Fleming Channel, upstream, midstream around Fighting Island, and then downstream around Boblo Island. And I did want to point out that we did actually, in fact, measure about 60 independent parameters at each station. You're not going to see all those today, but you know, I did want to point out that there was a lot, was an awful lot, and a lot more than even made it into this presentation that was done. So I mentioned we did a variety of statistical tests to look at these nearshore, offshore gradients and upstream, downstream gradients. I'm going to focus here on the two-way ANOVAs and the exponential regressions. The planktonic communities, they're typical biological communities. They respond to the seasons at, you know, the same way that everything else that's alive does. So they're very different in the spring, summer, fall. So we use the two-way ANOVA because we wanted to account for those changes and not just say, well, it's different in the summer and fall. That's, you know, that's not an impairment. That's just what it, you know, it's just what it does. So we wanted to make sure we accounted for that. And we also did some exponential regressions to look at upstream and downstream differences, which I'll come to as well. And if you're really interested in stats, you can read the section in our report on with the stepwise regressions, but I'm not going to get into that here. So really, there, in terms of looking at differences in the nearshore and offshore, there are very few. In fact, in terms of nearshore and offshore, where there was no seasonal gradient, we found the differences were in flow rates, light levels, and chlorophyll A. If you've ever been out on a boat on the Detroit River, you already know how different the current is nearshore than offshore, right? That's just showing that. That's all that is really showing, and that's consistent throughout the year. You've probably also noticed that the water is more clear offshore than nearshore. Again, there's nothing staggering in that. You know, and then chlorophyll A is, you know, a measure of phytoplankton biomass. And what that shows, you see higher levels in the nearshore areas than offshores throughout the year is really all that's saying. And that comes up too when we do the ANOVAs and we factor in and when we look at the phytoplankton, you know, in terms of what was significant, where a month where there was a seasonal gradient as well as a habitat gradient. Phytoplankton biomass was, of course, varied with both nearshore and offshore and seasonally, and that's entirely expected and entirely predictable. Within the phytoplankton community itself, it was primarily the grouping, the diatoms, that showed this change. The other groups had much more seasonal variability than anything, and we couldn't separate that, the seasonality from the nearshore or the offshore. And consistent with this difference in phytoplankton and chlorophyll in the nearshore versus the offshore, we also see that the photosynthetic rates, the primary productivity, is higher nearshore and offshore. And given that the current is reduced in the nearshore and light levels are a little bit more optimal in the nearshore, that's really, you know, what you would really expect to find there. So that's really it. Moving on to looking at upstream and downstream differences, we did find a significant difference upstream and downstream for zooplankton. as well as for zooplankton being lower downstream than upstream. And that's a fairly expected result for a river. We see that H&F is actually higher downstream than upstream. So H&F, or heterotrophic nanoflagellates, can compete with zooplankton for food. They eat bacteria and APP and picoplankton specifically. So when you find bacteria is reduced, you often find that H&F does a little bit better, because they're so that's a fairly expected result. We found silica, another nutrient, was slightly higher downstream than upstream. I think the difference was 1.61 versus 1.57 milligrams per liter. It's very minute. It's certainly not of an amount that would have an effect on the biological communities. Silica is important for certain types of phytoplankton, for diatoms in particular, and at that difference, it's not having any sort of meaningful effect on the food, on the diatoms, and hence on the food web. So those are really all the, and then I'll go to mention the exponential regressions. It's this graph at the bottom, the zooplankton, that you want to pay attention to. I included the Niagara River, that's this line in brown, in this graph as well. So this is really a very predictable rate of decline of zooplankton biomass along the length of the river. It's a very well-established relationship in the literature. It's seen in Europe and North America and really all over the world. The Detroit River looks a little bit more like a straight line, but that's really a question of scale. You actually are seeing that same basic exponential decline in zooplankton biomass from upstream to downstream. The difference is due to the fact that in the case of the Detroit River, we're really starting, 100 kilometers downstream of the source. The Detroit River is really the lower third of a connecting channel between Lake Huron and Lake Erie. So there's already been a lot of reduction in zooplankton biomass happening upstream. But that relationship is still there, and that relationship holds, and that's very much an expected characteristic of rivers. So we come back to our assessment criteria and what is happening to the planktonic communities within the AOC. Well, we see that we have higher phytoplankton biomass and higher primary productivity at nearshore sites. So there's more biomass, there's more photosynthesis. And we see lower zooplankton biomass downstream and then upstream, and higher heterotrophic nanoflagellate biomass downstream than upstream. So the key question, are these consistent with expectations for a riverine environment? The short answer to that question is yes. These differences that we teased out are very much expected for a riverine environment. So I'll wrap this up. So of course, the BUI is about phytoplankton and zooplankton populations, and they're linked very much to bacteria, to the microbial food web, to bacteria, picoplankton, H&F, and ciliates. We did this study during the spring, summer, and fall of 2019, so we wanted to very much capture the seasonal variation in all of these things. We observed very few differences in the planktonic communities that really were well explained by riverine ecology, which is to say there's really no evidence of anthropogenic stress causing those differences. And when we consider the planktonic food web, or the carbon pool, it was dominated by autotrophs, and it's very different from what we see in the other impaired AOCs. So with that, we concluded that there really is no evidence of anthropogenic impairment of the phytoplankton and zooplankton populations. So thank you for your time and for listening to me. And if you have any questions, I'm happy to answer them.
Thank you, Mark. So as Mark mentioned, we're now going to move ahead with the question and answer period of this open house. So as a reminder, everyone's video and microphone will still be muted during this time. So please type your questions in the Q&A box and we would be happy to answer them. Let me see here, Mark. Okay, so question, are smaller organisms more productive in the Detroit River, or is it the larger zooplankton?
So smaller organisms in terms of phytoplankton, yes, would be more productive in the Detroit River. So we definitely see more of them in terms of their biomass. So they do generate more.
That makes sense. What are potential implications of low zooplankton populations in the Detroit River for the food web?
Well, I mean, if there's not enough zooplankton, there's, at the core level, if there's not enough zooplankton, then there's not enough food. But are these, I guess, I'm sort of cautious in answering that question. I mean, I know the levels are low, but are they unnaturally low or absurdly low? I don't think that's the situation. Kelly Bowen, who is one of the co-authors of the study and our zooplankton expert, I know she has said that one of the difficulties with zooplankton is you're measuring what's there. Sometimes when you see low biomass, it's because a good chunk of it has already been eaten. So they could very well be regenerating themselves, but you're not necessarily capturing them with static measures that you get under the microscope. I mean, one of the questions, and I think this kind of gets at the BUIs around fish habitat and fish populations, which is sort of the next thing to, I suppose, look at and consider. But I'm not an expert on that, so.
No, fair enough. I think you answered it perfectly. So, and you alluded to seasonal changes in zooplankton and phytoplankton in the river, and I was just wondering what that looks like. Are they more in this fall or?
Well, I can show you.
Okay.
I have a graph here. So, yeah, am I still?
Yep.
So looking at the phytoplankton. So looking at the phytoplankton, for example, so in the spring, you look at this block, you see this big block of orange. So that represents diatoms, things like cyclotella and organisms like that. And they're very common in the spring and the later part of the fall. So that's one example. As you get into the summer and the warmer temperatures, you see more of these blue-greens or the cyanobacteria. So you see them more here in July and in September in some of the sites. And you can see in November that, the composition, there's actually more of the green algae, the chlorophyta, as a bigger part of the community in the fall. So they change with the seasons. Different things. I mean, every little species is different. They have different pigments. They respond to conditions differently, and how they respond, it's kind of amazing. I mean, one of the fundamental principles of ecology is called the paradox of the plankton, which is when conditions seem to get harsh, there tends to be more of them, and so they change. They respond very rapidly to conditions, and suddenly certain things find favorable conditions, and they start to do well, and that happens throughout the year. And I mean, it happens probably every day, but we only got out there we weren't out there every day. I can move on to zooplankton. And again, you know, you see sort of very different patterns too throughout the year. You know, there's differences in biomass for sure, but in the spring, you see a lot of the black here represents zebra mussel villagers, which are counted in as part of the zooplankton community, but they're very active at that time of year, and there's a lot more of them. Mm-hmm. You see by the late summer, calanoids and bosminids are very much the dominant drivers of the zooplankton community. And that seems to hold into the fall as well, although the biomass starts to decline a bit.
Yeah, it's very interesting to see how quickly it can change over a relatively short period of time.
Mm-hmm.
So, another question, how quickly could anthropogenic stressors push phyto and zooplankton populations in the Detroit River back to impaired?
That's a good question. Because I'm not entirely sure the system was ever considered impaired, but more importantly, I don't, I want to be careful about how I say this. In terms of what would be impaired, I mean, it has to be an ongoing problem. Like, you could have a leak at the sewage treatment plant, but that would be temporary, right? You know, you could see that and you might pick it up. But assuming that, you know, the problem gets fixed, then the community will recover quickly. And, you know, in a case like this where we're out, you know, we went out once about every 6 weeks or something. You know, let's face it, something could have happened in that time that we didn't pick up. You know, I can't say that for sure, but if it were really impaired, this problem would have to be ongoing, and we'd see it throughout the year in different ways. And, you know, hey, we wouldn't be here now. We'd have, you know, our conversations back in March would have been like, whoa, there's something going on here and we don't know what it is, right?
Yeah, for sure.
But I think one of, you know, it would, you know, to be impaired, it really needs to be persistent conditions.
Yeah, no, that makes sense. And then one final question, unless anyone has any others. What are ways individuals living in the Detroit River watershed can help protect phyto and zooplankton populations in the river?
Sorry, could you repeat that?
Yeah, so what are ways individuals living in the Detroit River watershed can help protect phyto and zooplankton populations?
I think a good one is, you know, I think is it the Windsor wastewater treatment plant or Public Works that has that program about what to flush, right? Though, yep. That's very important. You know, that is probably, you know, that's the first thing that comes into my mind, as what an individual can do, things like that. And just little, you know, little things when you're walking by the river, you know, don't throw your garbage in there. It sounds simple and maybe even trivial, and you probably never do those things, but you know, you still walk by and see garbage floating there and whatever, beer cans and whatnot. So I mean, those are by far the most important things.
Yeah, for sure. And watch what you're dumping, no chemicals.
Yeah, yeah.
Sink and things like that. So, all right. So I see no further questions. So with that, I wanted to thank everyone who was able to attend tonight's webinar. We hope you enjoyed learning about the history of this particular beneficial use for the Detroit River. And again, we wanted to thank Mark for coming out and providing an overview of the great research that he's done on this topic in the river. So as a reminder to those who would like to download the technical report and fact sheet that we mentioned, or to provide additional comments, you can visit our website at detroitriver.ca. And so we are accepting comments on this beneficial use until the end of December, so December 31st, 2020. And so now we would like to get some initial input on your thoughts on the DRCC's recommendation to change the status of the phytoplankton and zooplankton BUI from requires further assessment to not impaired. So I will put up a poll again. If you can take the time to fill it out. So basically we're looking at which statement best represents your opinion on the status of the degradation of phytoplankton and zooplankton populations BUI in the Detroit River AOC. Do you agree with it? Do you not agree with it? Or do you not know? So I'll leave that up for a couple more seconds. Perfect. So thank you for filling out the poll. And once again, we can't thank you enough for coming. So we look forward to your comments and we hope you have a great night. Thanks so much, everyone.